so you can all see my face. All right, so this is the fourth and final talk from me. This is going to be an overview of omics technologies. We'll spend a little bit of dedicated time talking about how SNP arrays work, and then also some of the technologies that are a little bit adjacent. And then, as I mentioned in the previous lectures, I spend most of my time working on RNASeq data, so we'll devote just a little bit of time speaking to how that is generated more specifically. All right, so we've seen this slide a number of times now. We've talked about the central dogma of biology, and we have the DNA piece, the RNA piece, and the protein piece. Obviously, there's a lot more components to biology that we're not going to talk about, but each of these has a corresponding omics discipline. So if you're studying DNA, you're doing genomics. If you're doing RNA, you're doing transcriptomics. And if you're looking at protein in the same kind of multiplexed way, you're doing proteomics. And so I'll say a few words about genomics and transcriptomics technologies. And then for proteomics, I'm just going to introduce it very briefly. As I've mentioned, this is a little bit of a divergence in technology from the other two. So we're going to talk about measuring variation in DNA first. And again, this is kind of going chronologically. So Sanger sequencing is the earliest kind of method of measuring this specific SNP level sort of differences. We'll talk about SNP chips, and then we'll talk about what people have been calling now for several years next generation sequencing. I would argue this is very much current generation sequencing, and it still was the last time we did this, Summer Institute. So this is becoming more and more just standard practice and less state of the art. So Sanger sequencing. I'm going to kind of stage this by talking about the Human Genome Project, which was completed in 2003. My slides did agree with the trivia answer without me changing them, which I actually wasn't sure that they were going to. And this was sequencing one haploid human genome. Notice that humans are diploid, right? So this is only one chromosome from one copy of each chromosome from each person, and not both of them. It took 13 years, and it cost almost $3 billion. This is from financial year 1991, based on how it was funded. So Sanger sequencing was the technology that was primarily used to complete that project. And I think we'll understand why it took so long and why it was so expensive when we walk through how Sanger sequencing works. So all of these sequence methods are going to use some component of that complementary base pairing mechanism that we've talked about in the previous lectures. And they're going to use that to determine some unknown sequence or some unknown base. So this was the first automated procedure, and it was based on a technique called, I don't even know how to say this, but I can explain to you how it works. I've always heard chain termination sequencing, and it was developed by Sanger, the company, which is where it gets its name. So we're actually going to walk through this diagram in a couple of pieces. So if this is the piece of DNA, the strand of DNA that I would like to sequence, the scientists will need to design a primer that matches some piece of this. So if you see something in a protocol that says random primers, they mean is this is the primer. This is the piece that has to match complementarily to some other sequence. In this example, it matches at the bottom, just to keep us all oriented kind of in the same direction. So this piece is going to stick onto this piece, and then they're going to toss in a DNA polymerase, and that's the piece that matches up the bases, along with regular old bases. This is just a different way of representing that same kind of monomer structure where we have the phosphate backbone attached to the sugar and then the base kind of hanging off the end. And then also in the mix, they're going to mix in these dideoxyribonucleotides. Kind of a mouthful. These are modified in two ways. One, they're going to have kind of a fluorescent tag. They're going to be marked and labeled with each base. So you have a G, a T, a C, and an A, and they're going to be differentiatable from each other. And then another one is that you're missing an oxygen here. And it turns out that the DNA polymerase needs that oxygen to continue past the last base. So how that all works in action is what's going to happen is in some cases, you're going to have the primer binding to the sequence that you're trying to sequence, and then you get one of these funny ones right away, and then it stops. And so you have this one thing hanging off the end. In other cases, you might have gotten regular bases all the way up until the last one, in which case this is an A. And so we're doing this in parallel with lots and lots of sequences. And so then you can arrange them from the shortest to the longest chain. And so you have your primer, which hopefully you know the sequence of your primer. And then this is the new sequencing coming up off of here. And the things we're going to be able to read are these last ones. And hopefully you have enough starting material that you can actually arrange these, if you arrange them by length, to get the whole sequence. So all of this is put through a laser, and in theory, you should be able to read the bases one at a time. And so the laser, between the laser and the detector, it's able to distinguish between these 4 different signals. So ideally, if you see output that looks like this is starting to look like nice Sanger sequencing data. And so you're seeing these peaks of green, red, black, and blue, and this is the sort of conventional coloring for them. And then when we talk about going from this kind of wave-looking stuff to actual bases, that's what we would call a call, kind of going back to our discussion from before lunch. So this all looks well and good. This is what a perfect Sanger sequencing output might look like. Also common is you might get an imperfect looking output. This is still real data. This still came from a real DNA sequence, but digesting this and figuring out which bases go in which order is certainly much more difficult here. So as you might imagine, the bases as they come off the sequence are assigned a quality score, and sequences of poor quality get resequenced. So you can imagine if we didn't know anything about the human genome, trying to get all the pieces and then putting them together would be quite a large task, which is what this picture is meant to represent. And this is just a cartoon. Let's say I have these sequences and I want to line them up. Trying to actually match base by base is a bit like doing a puzzle with no picture on it. Right? You can't really use the relative kind of context. You just have to go with what overlaps and what doesn't. And so this hopefully gives you some appreciation of why the Human Genome Project cost so much money and took so much time. So as you might imagine, SNP arrays were transformative in this space as well. We talked about microsatellite markers, but even if you could sequence something, it was very expensive and very time consuming and often limited to a really small region of the genome. So if you're now changing to a genotype SNP array universe, this is the same slide that I showed this morning. So we're just saying we're not looking at just these locations and not necessarily these consensus sequences that we mostly share. These are super cheap, we know that, but getting into how they work, this is also going to leverage that same idea that that complementary base pairing is going to happen somewhat naturally. So we start with 50-mer single-stranded RNA probes arranged on a chip. So they're just sort of sticking off the chip, and they're single-stranded, so that they're going to want to pair up with something. Some technologies also use DNA. So the single-stranded DNA from the sample binds to the probe, right up to, but not including the SNP location. So that's illustrated here, right? Where if this is our SNP and this is the probe, we want to match the sequence right up to it. Unfortunately, SNPs aren't super densely packed, so 50 bases is plenty enough to identify this location. And when it's not, we would call that bad probe design. And so this leaves the SNP base without a match. And you can take these same probes here and just allow them to be added by the DNA polymerase, and then this will either flash. I mean, usually it's red and green, but but it'll release the fluorescent probe and you get a little burst of light, and a camera can capture that. And there you go. So, yeah, go ahead.
Are we constrained a bit though by how, like, if there were two SNPs that were very, like, within 50 of each other, like, it's not a probe design issue, that's just like how would you get the one that's...
You wouldn't. Or if, conversely, if these sequences were very highly redundant, say you had some kind of homologous sequence elsewhere in the genome. And so it's probe design, but it's also like array design, right? That you want to choose SNPs that you know you can measure well. And so there are absolutely SNPs in the genome where if there's this SNP here and 5 bases and another SNP, another common SNP, that would interfere with how well you could capture this probe. Keep in mind that you can also go from both directions. Right, this gets into some of the quality control issues with like AT SNPs and CG SNPs. That one company might design their probe to approach a SNP this way, and then someone else might be targeting the other strand and go at it the other way. Does that help? Okay. So these arrays, you end up getting a whole bunch of these probes at a known address. So this is a cartoon array where we have 3 SNPs on our array, and we have each of these having little probes sticking out here, and then our sequences bind to them, and the fluorescently labeled tags pop onto these. And so then this marker on this particular chip would be fluorescing red, this one green, and this one, with these particular fluorescent markers, you get yellow when you get one of each, like approximately equal. If you do this across a whole bunch of samples for the same SNP, you could imagine plotting some measurement of the intensity of one of the colors versus the intensity of the other color. And this is when we talk about SNP calling, this is what we're doing. After we have all of our data, knowing that the SNP should be polymorphic, we're taking all of the ones that kind of cluster like they're mostly one color, all of the ones that look like they're mostly another color, and then everything that looks like they're halfway in between, and making a call based on that information. This is a very nice example of what it should look like when your SNP is of good quality. This is something that also happens. And this actually happened to me when I was working on a GWAS where we were trying to combine data from 2 different chips made by 2 different manufacturers. And some of the SNPs ended up looking kind of like this, where they would look really nice and polymorphic on the other chip. And so those were SNPs that we had to take out of our analysis because they were clearly not concordant between the two manufacturers' arrays, and they were going to confound our results pretty extremely. So this is where making sure that your minor allele frequency makes sense, that the SNP is not completely monomorphic, kind of comes into play, especially if you're comparing data across platforms.
Wouldn't it look like this though if it was like kind of rare?
Yeah. If it was super rare and we didn't have any minor alleles, you would also get something like this. And the only reason we caught it in the example that I gave was that we had 2 different chips. On a couple of overlapping individuals, those were not concordant, and then in addition, they were very similar populations. And the SNP was just always homozygous reference on one platform and not homozygous reference on the other platform, and that's how we found it. This wouldn't mess you up too badly if you had, say, all of your data collected on one platform and this just looked like it was a monomorphic SNP. It just contains no information, and so you can't get a significant hit from this SNP. Okay, so sometimes there are more than 3 clusters. I added these little clouds where they might be. This could also be indicative of some kind of artifact, but it could also be a copy number variation. If you imagine if you had an extra copy on one of your chromosomes, then you might have 2/3 green and 1/3 red, or 2/3 red and 1/3 green. And of course, the 3 of each case doesn't give you any information because you just land in one of these clusters. But if you start to see this kind of fan shape, this could also be a CNV, and I know there's quite a bit of research going on about how accurately could we call a copy number variant from array data. Yeah?
Is this desired?
Sorry?
Is this desired? Like,
it's desired if you want to call copy number variants. Like if you're specifically interested in probing copy number variants, and if you want to be able to associate them with phenotypes, then yes. This is not desired if you want to make a standard biallelic SNP call.
Because I see in the cartoon each SNP has 5 probes, so yeah, if they only had 2, then you wouldn't have to worry about this, right?
Yeah. Okay. So this is kind of the basics of how SNP arrays work. And so if all works as planned, you actually get your data back, and you can have genomic locations on one of your axes, and you can have samples on the other axes, and then you can just have a matrix of genotype calls, which is very convenient and lends itself to a lot of the kind of standard statistical procedures that we're familiar with today. And so that's how this technology became just extraordinarily useful and extraordinarily inexpensive and powerful way to query this kind of data. So Dan actually showed this graph in his intro lectures, but I think with this slide we're going to start talking about next generation sequencing. So if we have any lingering questions or thoughts about SNP arrays, let's have them now. Yeah?
I was wondering whether the comment that you used the SNP arrays like in a segment of like the genome that has been like, or is it the entire genome most of the time?
So they normally try to put the SNPs across the genome like, sorry, for the folks on Zoom, the question was how are the SNPs distributed? Is that an accurate representation?
No, like the segment of like the genome that we want to sequence, like a genotype. Is this mostly applied to the entire genome or to maybe an amplified segment of like, the entire genome?
So it depends on the chip. Most manufacturers will offer kind of a catalog of chips that you can order. Most of them try to at least tile the genome, kind of like mile markers on a highway. So you have a SNP at least every so many bases. In general, centromeres are going to be hard to get, just because they do tend to be all gunked up, and then by the time you get all of the epigenomic stuff off of them, sometimes there's not much DNA left. But in general, you try to tile everything, and then some of the custom arrays can target psychiatric phenotypes or metabolomic phenotypes. And there are a lot of arrays that try to more densely populate the arrays in places that you might care about. And most array platforms will also offer a custom option. So if you say, I really want you to cover everything you can in these 10 genes, they'll do their best to accommodate you for, of course, a price. Yeah.
Maybe a lot of the data that guides the array design is coming from European samples.
That's my understanding of it. I haven't done a whole lot of research into that, but that is my understanding, that a lot of the array designs are done based on European samples and not on global samples, and so the SNP choices themselves are fairly Euro-biased. Dan?
I think there's a new one called like the global SNP array or something.
Oh, cool.
Supposed to be more representative of other populations.
That's awesome. And I would guess probably not the first iteration, that it won't be the first iteration of such a project as more sequencing happens. Okay. Any more SNP array stuff? Cool. So next generation sequencing, as I said, this is really more like current generation sequencing, is used in really a lot of different contexts. And this is getting at the figure that Dan showed on the first day, where we have Moore's law, this kind of almost an expectation of how quickly we can expect the cost to drop off. And this just incredible dive down, and this big crook right here is the advent of next generation sequencing. And so it's got a lot cheaper and a lot more parallel than Sanger. And it takes advantage of alignment. And this is a piece that we'll talk about as well. So this is a schematic to demonstrate how next generation sequencing can work. This is specifically how it works in humans. We cut the DNA into overlapping fragments that are short enough for this type of sequencing. That length depends a little bit on what technology you're using, but it's in the hundreds sort of order of magnitude. We clone the fragments in a plasmid or in another vector so that we have more of them. This keeps us from having to collect tons and tons of DNA. And we sequence each fragment one at a time, put all the sequences into one big sequence with computer software. And again, this can be done in the style that I mentioned before, where it's kind of like a puzzle that has no picture. Or if you have a reference genome to use, you can use the picture or the reference to help you put things where you want them to go. This is another schematic that I'll get at the how of the sequence. So you start with genomic DNA, it gets cut up, we already talked about that. It needs linkers in order to interact with the flow cell, which is the piece of technology that this uses. These linkers then bind to locations on the flow cell, and they end up making these like little rainbow type structures. So you have the flow cell holding on to the molecule that you're trying to sequence on one side with one linker, and on the other side with another linker. Sequencing then proceeds very similarly, where we're using a polymerase to match the bases, but now we're doing them in sequence without stopping. And there's a camera that's watching the colors change and recording them. And so part of this technology getting cheaper and faster has to do with this optics component, that as the cameras got better and more accurate, you could cram more of these little tags onto a single flow cell and thus get more sequence out. So this is what they call real-time base calling. So if this is my sequence, you might watch a single dot and you might see the colors change from green to blue to red to green. And the computer associated with this very tiny camera is reading green, blue, red, and translating this into sequence. Another example would be the sequence on the bottom. That's this little dot. And you can see that it stays green for several cycles. And so you're just reading T's until you get a G, and so on. And so this technology was transformative in and of itself, just that you can sequence this many bases this quickly, that we're not relying on a different segment of DNA to call each and every base. But also, I think a huge help here is that we have done the Human Genome Project, and that these fragments can then be aligned to a reference genome. So this allows you to determine where each piece of DNA is located in the genome by arranging it to sequences that we already know about, that we've already seen. So if you're trying, this is what you would call assembly. If you're trying to assemble the pieces that you've sequenced, just based on the sequence that you see. And then if I know that I'm trying to make a picture of a tree with some grass and there's some sky up here, that it might help me rearrange these pieces more quickly and more efficiently. And the same is true of the reference genome. Of course, we are not all the same, so we don't all match the reference genome perfectly, but it's often enough to give us an idea of where pieces might fit together. So this is a really common visualization for next generation sequencing data if you are dealing with the level of reads. So this would be the position on the chromosome. So this is the reference genome up here. And then these are your reads that have been aligned to the genome. And you can see there are little places where they're different. Sometimes these are sequencing errors, and sometimes these are actual SNPs. And so if this is from one sample, we see these little blips, and just these little like kind of one-off moments where the sequence appears to be different. The aligners don't get too hung up on that. There are error adjustments built into these models already, but sometimes you see a place where about half of the reads have a reference. They're gray, there's nothing happening, and then about half of the reads have a different base there, and they're all the same. And so this is something like a SNP call in next generation sequencing. We also know where to look for a lot of common SNPs, and these do tend to line up. So if you were looking at your next generation sequencing data and hoping to call a SNP, you might infer that there's a SNP in this individual at this location. All right, so as you might imagine, it depends on how much sequencing you do. If you only sequence a couple of reads in a region, it's going to be really hard to tell if you just got unlucky and didn't see both chromosomes, or if you're actually seeing the true genotype. So the number of independent sequencing reads that cover a particular nucleotide is referred to as coverage or sequencing depth, kind of depending on your application, but for many applications, these are used interchangeably. And for a typical whole genome sequencing project, coverage ends up in the 30 to 50x range. This is increasing as things get cheaper and people want higher quality data. The deeper the coverage, the more confident you can be about your genotyping calls, which makes sense, right? If you have more reads and you can confidently say that about half of them look one way and half of them look another way, it's very easy to make a call. Fewer reads, it's harder. And then obviously more coverage means more money at any fixed point in time. So that's next generation sequencing for genomes. I'm going to talk about exomes next, unless anyone has questions about genomes. Patrick's making the face that looks like he might have a question.
I was just thinking about low-pass sequencing, but I don't know if it's...
Oh, yeah. So yeah, that's actually worth mentioning. So low-pass sequencing can be really powerful if you're interested in population-level inference and not necessarily being able to talk about individuals.
I think, isn't GenCode's thing that like they do low-pass sequencing, but then impute to like solve any errors? I don't know enough about this to,
Gotcha.
to say much, but
Yeah.
Dan knows more than I do.
My understanding is exactly what you said, Patrick. That's the plan. The issue is it's less useful than SNP arrays when you don't have very much data, when you don't have enough individuals. But when you have a large enough number of individuals to be able to do good imputation, then it's more powerful than SNP arrays, because you have you get like 40 million SNPs instead of 10 million SNPs.
Because yeah, you could have very low, 5x coverage, and there'll be errors, but then the errors will be fixed by the imputation, at least for common variants.
They're not really even errors. They're just missing, or like.
But sometimes also sequencing.
Sure. I think I was thinking more in terms of like if you do enough people, and you don't cover every place in the genome with every person, that's okay, because you're still getting a random sample of the genome, and you have tons and tons of people, and so in theory you would get a good sampling of the common variation in that region. That part makes sense to me. I'm curious about what you mean by impute.
Me too.
Okay, we'll take that discussion offline. Grant.
Is NGS more accurate than an array? In terms of SNPs, at least.
I think it depends on your array and it depends on your coverage. So at low coverage, absolutely not. For an individual base, I think it's still less accurate.
NGS is less accurate.
Yeah.
Okay.
Because the SNP array is designed to capture that one base. And it's asking you, is it A or is it B? And not, what is the base here, right? So the question is a little bit easier, and the design has been to just answer that one question. Tammy.
You mentioned that centromeres are difficult to capture with SNP arrays. Are they easier to get with NGS?
That's a great question. I'm not actually sure, but we can look into it.
Isn't the problem there that's highly repetitive DNA?
It's tightly packaged and highly repetitive, and so often when you look at the reference genome, you'll see like ends through the middle of a chromosome, and I don't think that is necessarily ends as in they don't know what bases they are. It's just they don't actually know how long it is, and then you see that at the telomeres occasionally too. Cool. Okay. Shall we talk about the exome? Okay, so this has come up a number of times as we've been talking about basics of biology, that the protein coding of the gene portion of the genome, or the genic portion, constitutes between 1% and 3%, and this has certainly been hotly debated, especially in recent years as folks learn more and find more and more molecules in biology, but it is still a relatively small fraction of the whole genome. And this we call the exome because it is coding the exons. This encodes majority of the functional part of the genome. I almost want to put scare quotes around functional, but you know what I mean, central dogma-wise. And so the annotation for which sequences encode protein is periodically updated, but usually these technologies can keep right in pace with that. This is much cheaper to sequence because you're not sequencing the entire genome, you're just sequencing a little slice of it, and this was a really nice little graphic from the University of Washington that I borrowed, whereas if you think of an apple being the whole genome, it's certainly more to eat the whole apple than this little tiny slice. So exome sequencing does require a little bit more work up front, that you have to figure out which pieces of the genome you want to capture before you can go and capture them. And so this generally will capture between 25 and 60 megabases of sequence at a time. And this involves in-solution DNA probes that bind to short pieces of sample DNA, and they're designed to get right around the genic region, kind of like parentheses, where you're trying to capture the gene just so and slice out that part of the DNA and then put that into your next generation sequencing example reaction. So they target the start of coding regions and then sequence a specified distance from there is kind of another way to think about it. So if this is gene 1 and this is gene 2, and these are their exons shown here, you could imagine that you do some fragmentation and then you have lots and lots of bits. These are sequenced exactly the same way, where you put them on the flow cell. And this is generally done with one end and not two, but there are folks that'll do it with two ends. You have the captured DNA, and then you have the captured DNA in solution, and it ends up getting sequenced. So you can also focus on a panel of specific genes. So if we could target everything that gets coded, you could imagine, oh yeah, Dan.
Just to understand the motivation for the exome sequencing,
Mm-hmm.
Is that what you would want to do? Is the main reason you want to do that because you want to study rare variants and you don't want to spend the money on whole genome sequencing?
That's certainly one of the primary motivations that I've seen it used. Yeah, that if you, certainly in rare diseases, it comes up in medical diagnoses as well. So folks who don't have a diagnosis, and we have either all the genes or a handful of genes that might be affected, nothing's coming up on a SNP array, you do whole exome sequencing, and if there's a problem in a coding region, you would see it right away.
Are there other uses that you've seen?
I've definitely seen larger panels of exome sequencing, sort of with the same logic as the low-pass sequencing, where you're just going to sequence a whole bunch of people and hopefully gain more power by having more samples as opposed to more sequence information. And then hope that whatever signal you're looking for is in the data that you've collected or somehow an LD with it, so that you can get some kind of signature. But I think the one you mentioned is the main one. Cool. So if you can slice out all the genes, you could also slice out just a couple of genes and save even more money or do even more sequencing on just those genes that you care about. This reduces cost, obviously. So you could look at, say, only genes that have been associated with autism. I've seen this done for only genes associated with muscular dystrophies as well for undiagnosed patients. The goal here would be to design your experiment to have the lowest cost and the best data quality for the question that you're answering. So if whole genome coverage is normally 30 to 50x, whole exome coverage is often 100 times or more. And then if you wanted to do a really targeted gene panel, you could get up in the 300 range for about the same cost. And so that's sort of the idea behind these targeting sequencings as I throw things.
Is the benefit of coverage to catch mistakes? Like, I'm sort of confused on why you would want to spend money resequencing a nucleotide.
Gotcha. So I'm coming from the RNA-seq side, and we have a different answer to that question. But for my understanding is it's one, to catch mistakes, two, to make sure that you're capturing variants accurately, whether it's a SNP or an indel, CNV, all of these things that can go wrong or just be different, I guess. Are you confident that that's a real call or a variant? So it's not necessarily to catch mistakes in the consensus sequence, but to be able to tell variant call apart from error.
Okay.
Does that help?
I think so.
Okay. Yeah.
Is it the case that going from, say, 50x to 300x is perhaps more beneficial for things like indels, but less beneficial for things like SNPs? I'm just trying to understand.
Yeah, that intuition tracks. Yeah, like if you don't need 300 coverage, why would you do it?
Right.
Kind of an idea. I would say probably you wouldn't. You would collect more samples or you would spend less money. Raymond.
I think the other instance is for somatic variants, especially in cancer.
Yeah. That makes sense. So the question was, why would I do this? What are the benefits? And Raymond's additional point was that you could also catch somatic variants, so if some proportion of your cells had some kind of variation and you were trying to catch that. All right, so there are a lot of other applications for next generation sequencing. My favorite is definitely RNA sequencing, which I'm going to walk you through a little bit here. There are ChIP-seq experiments where you can only sequence the RNA that's bound to a specific protein or a specific molecule that you're interested in. Methylation sequencing, or methyl-seq, this assesses the sites of methylation, so this is very relevant to epigenetics work, and also a data type that I've gotten to work with in the last year, RiboSeq, which assesses which sequences are bound to the ribosomes. So if you're not sure if you can do proteomics and you want to know what's being translated, you might look at what's being bound to ribosomes instead. So I'm going to talk a little bit about RNAseq and how that works. It's obviously going to be really similar to DNA sequencing with the same technology. There are going to be a couple of crucial differences, namely around what kinds of questions we can answer. So as you might have already inferred, next generation sequencing can be applied to RNA as well as DNA. And we first start by making cDNA from the RNA by reverse transcription. So this is something that retroviruses do, but humans don't do internally naturally, that we take RNA and we turn it back into DNA. It's sort of like decoding the message. We do this because the cDNA is much more stable. So the RNA can provide us different information about the biology, specifically which genes are active, what's differentially expressed between conditions, where are the splice junctions, and what combinations of splice junctions are present. As you can tell, I'm a little bit splice junction biased. So this is going to be a cartoon. This is going to animate kind of this whole process. So this is for the first application of RNA-seq, which is transcription quantification. So if we have our genomic DNA up here, and we know from the central dogma that this encodes RNA, so say these are all our RNA transcripts, getting rid of isoform diversity for the moment. We have many transcripts in a sample. We might want to read these guys. And so the Zoom thing is a little bit in the way. But we might want to grab these guys. In general, messenger RNAs have a long tail of just A's at the end of them, often called the PolyA tail. And so this would be a method called PolyA Pulldown, where you'd have a bead with sequences of lots of T's on them. And ideally, your messenger RNA would just glom onto it. There is the possibility for breakage and losing information over here on the 5' end of the transcript. But nevertheless, you have some captured fragment of RNA that can then be digested into fragments. These digested fragments then become cDNA. This is that backward sort of decoding thing. Again, humans don't do this naturally. We don't go from RNA to DNA, but retroviruses do. And then make more of this so that we can sequence it. And very often RNA-seq is done with this kind of rainbow structure sequencing where you have reading from two sides. And this helps a great deal in putting it in the puzzle and into the reference where it goes, especially because you're navigating splicing. So this is a little schematic just summarizing what we just said. We got paired end reads out the bottom. And then we don't know where these align to the reference genome, so we have to align them back to the reference genome. Again, accounting for the fact that we might have splicing in there, and so we might have big gaps. And some of those we will know where they are, and some of them we won't. So you can imagine having done this a whole bunch, and that these are our paired end reads, and this is where they align to our reference genome. You can get some idea of where in the gene they land, but you can also just count them up. You can say, I've measured gene 1 this many times, gene 2, gene 3, and gene 4. And so now this gives you some kind of quantitative idea of how much expression of different genes you have relative to one another. So this is starting to get at like which genes are active, which genes are differentially expressed. Of course, these quantities will need to be normalized across samples so that you can actually use them in a meaningful way. But this is just kind of intuitively how we go from molecules and flow cells and fluorescence to data we can use. Okay, so this is a topic very dear to my heart, is DNA or RNA sequencing for splicing. So if we zoom in even further and we have, say, one gene this time, but now I've colored the exons 4 different colors. Again, this is just a cartoon gene with 4 exons. You can imagine that we might have a couple of different isoforms present in our sample and chop these up just like we do with anything else. We end up with fragments for sequencing. These sequencing may or may not include a junction between exons. But if they do, they contain unique information about splicing. So if we take just those reads and we paired and sequence them, again, some of them will have the junction actually captured in the paired end sequence. In some of them, you'll know that there's a piece of the blue exon and a piece of the red exon, but maybe the exact location of the splice junction is lost. And so naturally, you can get a lot more information from this than you can from this, but they're still both useful. So if you take all of these and align them, because remember, we don't actually know where they came from when they come off the sequencer, you have to align these back to the reference genome, or in some cases, the reference transcriptome, which incorporates some of the annotated information about where these splice junctions fall. And the more sophisticated aligners now will let you let the algorithm discover new ones. Yeah.
So you have these little H guys.
Yeah.
At the top, right?
H guys.
And so there's a line in between them.
Mm-hmm.
So is each arm something that you sequenced, or like because you said you broken
Yep.
broken it up into little pieces. I'm just trying to understand. Is like one unit a piece and you've just not read the middle?
Yes, that's exactly correct.
Oh.
So this represents a paired end read. This is the rainbow kind of sitting on the flow cell. Now the rainbow has been straightened back out, and we've sequenced a bit on this side and a bit on this side, and we can try to kind of infer what happened in the middle, but the polymerase didn't actually sequence all the way through. If you get a short enough read, sometimes they'll overlap. This is explicitly modeled in most aligners. Insert size or gap size is often what they'll call it. But yeah, sometimes the segment was so short that the reads actually overlap.
So I thought this was using the same technology as the next gen sequencing. Doesn't next gen give you the full readout on the whole rainbow?
It depends on the technology.
Oh.
So some of the next gens will use a paired end too. It's just if you look at like an IGV snapshot, geez, I have a lot of animations in here. Okay, so if you look at something like this, often the little arrows indicate which side of the read you're looking at. So, and both the RNA-seq sequencing and the just normal DNA sequencing can be single end or paired end. But yeah, this visualization will not show you the middle. Like if you click on one of these reads and you say, show me the mate pair, sometimes it'll show you.
So I'm trying to understand this picture then. So you have these gray bars, but really the only thing that we're actually sequencing is stuff near the edges where the gaps are?
No, all of these guys have been sequenced. This is like a super zoom in and squish down of the H guy. So this would be like one side of the H, and then the other side might be somewhere over here.
Oh, I see. Okay.
So this is much more zoomed in than the RNA-seq example that I was giving. Does that help?
Yes.
Sweet. Okay. I'll quickly relive this animation. So we did that already. So as I said before, some of these will contain information about the splice junctions, which could be interesting in biology if there's an error in splicing or if there's, oh yeah, go ahead.
I was wondering whether like RNA shearing is happening at specific intentionally at specific sites, like maybe using are we using nucleases like to shear?
It depends on the protocol. Some of them do, some of them don't. So some folks use sonication, like hit it with like a sound wave and supposedly breaks up randomly. There are just a number of approaches to that. So sometimes.
Andrea?
Yeah.
Sorry to take you back to this, but
No, that's okay.
why again is it that you get the you only get the ends and not the middle, and how is it that they end up getting paired?
Gotcha. Okay. So they end up being paired because they were all the same piece of DNA that we were trying to sequence, and they don't all end up getting sequenced. And again, this is my understanding of it. I hopefully reserve the right to be incorrect, but my understanding of it is that the polymerases kind of poop out. That getting a polymerase that doesn't just kind of either disintegrate or stop working for a very, very long stretch of DNA is fairly difficult. And that's one of the places that the technology has been advanced. And so the kind of most powerful strategy to collecting this kind of data they've found is to sequence, because you could just sequence 100 bases and then try to put them all together. And you could sequence shorter pieces and try to put them all together, but of course that's harder. And what they found is that it was more powerful to sequence 100 or so bases on one side, have a gap that follows some distribution, and then sequence another 100 or so bases. And so this was a lot of, at least my understanding is inside of the industry, it was a lot of trial and error and finding how can we minimize the amount of sequencing that we have to do in order to get uniquely aligning reads.
The way you get the two ends is that you're going in both directions.
Yeah.
How do you know when you do that you're how to match up the
Okay. That's done with the bar coding. Where is this picture? There are adapters on the piece of sequence. Where'd they go? Like, how do you know which one goes with which one? Yeah. That's done with these sequences. And it's also done with proximity on the chip. So they've made it so that one end has one sequence that binds to the blue probes here. And then this end has another sequence and that binds here. And this is showing them kind of all mixed up. But I think that in practice it's a little bit more deterministic. Like if your blue end binds one place, that there's a very limited number of places where the red end can go. Yeah.
I just want to clarify one more thing with the notion of it sequencing in both directions.
Mm-hmm.
Is what's meant by that is that you do one strand 5' to 3',
Mm-hmm.
and then the other strand on the other end also 5' to 3'.
That's a really good question. I don't know,
Okay.
actually, but I bet it's in the manuals for the manufacturers. I'll have to look into that.
Okay. It's not. I don't think it's like a particularly important detail, but I'm just confused if like, are you going 3' to like it does?
Like, do you do it backwards?
Oh, you can do that.
I don't know. It's remember that this, because you have two strands, one is going 5' to 3' this way, and then the other one is going 5' to 3' this way. So if you started on this side, you would just be sequencing the other strand.
Right.
If you were still going 5' to 3',
Right.
which is
I see.
which is my suspicion of what's happening, and then inside of the processing software they just flip it.
Okay.
But I don't know for sure.
Okay.
We talked about all of this. And we talked about splicing. And I mentioned that in order to get this information back, you actually have to do an alignment. And this kind of alignment has become mature now in the last couple of years. And these are quite reliable, even if they are not annotated, which is pretty cool. You can discover previously unknown splice junctions using this technology and the set of computational tools that are available. So we talked about RNASeq, we talked about all the different things that it can measure, with a little bit of an emphasis on splicing, because that is something that I work on. Measuring protein becomes a totally different ballgame. There are a number of ways to do this, and this is really getting outside of my expertise. DNA and RNA are linear. They encode sequence using 4 possible characters. Amino acid sequences have more possible components, but they also have many properties beyond their sequences, and these properties are not always immediately predictable from those sequences. This includes 3D structure, folding, which is kind of related, and forming complexes with one another. So this is an example of a protein complex where there are several different peptide strings all folded together, and this all needs to be folded together correctly for this to work. So the 3D structure of a protein is critical to the function, and sequencing a protein the way we sequence DNA and RNA is not quite so straightforward. So measuring protein in a multiplexed way often requires an antibody, and until relatively recently, this was often done one at a time, where you'd have one protein that you were interested in and a number of samples, and you would run them painstakingly, one protein at a time. This is still done in comparatively small batches of proteins, so many of the modern proteomics panels have thousands of proteins as opposed to all the genes. And then there are many types of measurements. You can look for the total amount, you can look at the amino acid sequence, or you can ask other questions about function and structure. I don't have a whole lot else to say about measuring protein, since it is really more removed from sequencing DNA and RNA. but I did include this little picture of how this might work. So you have an antibody and the protein that you wish to measure, and you have to capture it. But as you can see, this is starting to look like a very different process than we've been talking about before. Human molecular genetics will get a little bit into proteomics, but not as much as the stuff we have been talking about. These are still super useful resources for all of this material. And with that, I'd like to thank you guys for having me and for listening to me a whole lot. Okay. Should we break? Or are there more questions? Yeah.
Do you use long reads for the RNAseq?
Yeah, so I didn't talk about long read sequencing in this talk, but yeah, long reads are incredibly valuable, especially if you're interested in a gene or a handful of genes. Also expensive, but super useful for especially deconvoluting transcript isoforms. Like you can say we have great diversity in splice junctions in one area of the gene and then great diversity somewhere else, and figuring out what goes with what. Like long read sequencing has been invaluable for that. Go ahead.
I was wondering, given the way that we sequence the data, is there no way that, say later, that we develop the technology to read the entire genome with the samples that have already been collected? Is there any way to utilize that data, or do we need to collect new samples?
Oh, that's a really good question. I'm not sure, because I'm not sure what that technology would look like. Raymond, have you worked with our Danish blood spot data? So this is an example that's very akin to the question you asked. So there was a Danish researcher who kept heel pricks from newborns in Denmark. And this is newborn, just a little blood spot. And they finally were able to actually pull some genomic data out of it. But I don't know much more than that, and I know Raymond has worked on it. So maybe he could say a few words about that.
Started biobank collection, it was late 70s, and they essentially just sat in the freezer, and the technology was around to amplify it up and do SNP chips, so this was the medical application. There was certainly some degree of artifacting evident from the fact that they had been sitting in the freezer that long, and the necessity of amplifying up enough DNA to work for getting calls. But especially for common variants where like your prior is better, there is actually variants there to call, has worked fairly well. More problematic in terms of negative like CMV type coverage. But like so to the general question of like ability to go back to samples and apply new technologies, it's largely going to be dependent on what protocols in place for keeping the samples in some form of the biological sample, whether that's some reserved quantity of the blood was kept, or some reserved quantity of the purified DNA was reserved versus was run through on a chip or on a flow cell for sequencing. And that's going to be done. There's cost features there, obviously, because you're using freezer space and it's sitting there for a long time. So it's going to be kind of study specific in how they keep their samples.
Cool. Yeah.
Given how mature a lot of this technology is, are we getting to a point where like GWAS analyses are starting to incorporate more multi-omic data, or is that still a pretty rare thing?
I think it depends on the research question of the GWAS analysis. Certainly big consortia, especially if they're running something other than GWAS. Again, GTEx is always my favorite example, where they ran just tons and tons of bulk RNA-seq data on postmortem people from a number of different tissues. Every single one of those individuals has a SNP chip.
I see. Okay.
Some of them have whole genome sequencing as well, but every single person has a SNP chip. Dan.
I wanted to add one thing to what Raymond was saying, and then
Yeah.
ask you a related question.
Yeah.
So the health and retirement study followed the strategy that you outlined. They collected saliva samples from participants, and they felt it was urgent to do that because it was an older sample, and they knew that some of the respondents were going to die. But then I think they extracted the DNA and froze the extracted DNA for a few years. And then when the cost of the genotyping was low enough, they went back and did the genotyping.
Nice.
So that can be a good strategy if you'd like to genotype participants. If you run lab experiments, you can collect saliva samples and freeze them for a while. But Andrea, I wanted to ask you, do you know why the cost of this SNP array genotyping has been declining as quickly as it has, and can we expect, how long can we expect that to continue?
Yeah, I don't have a good intuition about that. I imagine it's just, at this point, it's incremental improvements on how much you can put on a piece of real estate. So the older SNP chips used to be very large, and now you get a lot more calls for about the same amount of space. I imagine optics probably plays a role, but beyond those kinds of speculations, I don't think I can tell you. But